Perfect. Thank you very much. Well, I'm assuming this is the afternoon in Europe, so good afternoon to everyone. So my name is Muta Rafi. I'm basically the VP R&D of Defense Therapeutics. For those that have not been following us since last year, this is basically our third presentation to give you an update on the research we've been actually doing in the last 6 months or so. So today what I'll be doing is I'll be just reminding panel, whoever is listening to what Defense Therapeutics does, and be focusing specifically on a new therapeutic anti-cancer vaccine that we are working heavily on and hoping to start a phase 1 trial early on next year. So again, Defense Therapeutics is a Canadian biotech company that is working on different products or different therapies targeting both immuno-oncology and infectious disease indications. So the entire company is really focused on a platform called the accum, which stands for the accumulator. And the core of this technology is that it can actually enhance the accumulation or delivery of biomolecules like proteins, RNA, DNA into specific compartments within the cell. And we'll go into this a little bit more in detail in the couple slides and in a few slides. And of course, the company is has this technology that is highly versatile in the sense that it's not only specific for one indication, but you can actually apply it to develop a variety of products that could target multiple diseases or indications. It has an attractive capital structure. And of course, what's really impressive about the company is that we are always achieving data-driven inflection points, I want to say every 6 months and so. And right now we are really preparing 2 phase 1 trials, that one in Canada and one in the US, and we're even discussing a third one that should be starting early on or by mid-2023. Now, what are we focusing on and why? I mean, everyone who's interested in the field of oncology knows that there's what we call standard of care. So standard of care is an approach that is usually encompassing, surgery, radiotherapy, chemotherapy, or a combination of these different therapies. But the problem is once you have patients that have reached a very advanced stage of cancer, let's say stage 4, where we have disseminated tumors all over the body, it becomes practically impossible to treat these patients. And therefore, we need to come up with new technologies or new treatments for these patients. Now, there are a variety of targeted biopharmaceuticals. As an example would be antibody drug conjugates that have been used to specifically bring in chemotherapy to cancer patients while avoiding, healthy tissues, cancer patients. And the idea is to kill really tumor cells without affecting healthy tissues or healthy cells. But the problem is these antibodies get entrapped into structures within the cells called the endosomes, and they eventually get degraded. Because of the acidification, they cannot be released, and therefore the drug doesn't end up really reaching the target site in the cell. And if you think about, developing, let's say, a cancer vaccine, so the idea would be to not only develop a memory response to protect the patient from subsequent challenges or, tumor growth. The problem is the idea would be to actually develop a vaccine to treat cancer. So you can kill the tumor and develop a memory. But the issue at the moment is that there is no real cancer vaccine available at the moment. I mean, we all heard about Dendreon, which have developed a vaccine against prostate cancer, but these data or the effect of this vaccine remains highly questionable because of the short improvements that they could trigger in patients. So there is really also a major struggle in developing effective vaccines against new deadly pathogens or emerging pathogens. And I'm pretty sure the best example I can give you is the COVID pandemic. It took us a while to develop a vaccine. So I think this is important for us to keep in mind so that we are prepared for future pandemics. So overall, there is really an urgent need to develop a variety of products to really answer various unmet clinical needs. So this is where we believe the accum technology comes into play. So the accum itself is very simple as a molecule. So it's basically a bile acid fused to a nuclear localization signal. So this is a small molecule of about 2 to 3 kilodalton. It varies in size because you can basically play with the bile acid component or the nuclear localization signal component. So you can develop libraries of accum variants. And so the idea is to really promote endosome to cytosol escape. Here's an example. So if, for example, you express in a given dendritic cell a construct composed of the enhanced green fluorescent protein fused to human galectin-3. So galectin-3 is a molecule that binds to glucose residues on proteins. And these proteins are usually found at the cell surface or within the endosomal lumen. So they're not really found in the cytoplasm. So if you express that construct, the entire cell will turn on green. So if you give, for example, an antigen to the cell will remain green. The interesting part is that if you deliver an antigen fused to accum, then what happens is that this accum molecule is going to start breaking down the endosomal membrane. So now whatever that was found on the cell surface and eventually got in an invagination process within this endosomal structure is now exposed to the cytoplasmic protein because the membrane is damaged. So whatever protein that was actually in the cytoplasm now will migrate to the endosomal lumen to be able to bind to glucose residues. And that's why you go from a cell that is entirely green to a cell where you start seeing puncti of concentrated green points. So these are endosomal structures that have been broken down. So this is a great example demonstrating how this molecule works. And if you think about it, this applies that this molecule could be used to the development of a variety of antibody drug conjugates, or even protein, as well as cell-based vaccines. Now, overall, the company has been heavily involved in developing a variety of products. So we've developed a variety of different protein-based vaccines. We're talking about a vaccine against HPV. We even have developed a vaccine against COVID. We worked through a variety of libraries to identify a new accum variant that actually shows potent anti-cancer properties. So this molecule could be used directly as an anti-cancer molecule. And of course, we have a variety of different projects where we demonstrate that the accum itself could enhance significantly the potency of any FDA-approved antibody drug conjugate. And that's important because it means you can actually apply this technology to any commercially available antibody to enhance by at least 100-fold the efficacy of the antibody, or you could use it to recycle anti-ADCs that have, for example, failed a phase 2 trial due to efficacy or failed because of dosage or dosing. But today what I'll be doing is I'll be talking specifically about another project we have, which is to develop an anti-cancer cellular vaccine. So these are different products we have at the moment in the development stage. Of course, the vaccine I'll be talking about today is the ARM vaccine, and this is because, and you'll know where the ARM name comes from. This is a vaccine that has actually completed now GLP studies, so we are really preparing our IND submission to be able to move forward into a phase 1 trial in Q2 of 2023. Now, what is the ARM vaccine? So this is what we call the A1 reprogrammed mesenchymal stromal cell vaccine. So ARM stands for A1 for the R and for the M of mesenchymal stromal cells. And this is a vaccine that is being used to target solid tumors. And what we have in mind to begin with is melanoma, but you can actually use it against breast cancer, head and neck, and any kind of non-Hodgkin's lymphoma, any kind of solid tumor that you can actually reach. So just a brief reminder of the only FDA-approved vaccine on the market. If you look at Sipuleucel-T, which is a DC vaccine, this is a vaccine that is composed of dendritic cells, and it's meant to target prostate cancer. So the idea is actually remarkable, and if you think about it, it's just generating DCs and pulsing these with an antigen. So you take, for example, monocytes from patients that you want to treat. You actually pulse them in vitro with a fusion protein between PAP, which is an antigen for prostate cancer, derived from prostate cancer, but fused to GM-CSF, which is a cytokine that will drive the monocyte differentiation into a dendritic cell. So the idea there is that the PAP will be taken up by the dendritic cell, processed and presented on both class 2 and class 1. So these are molecules on the cell surface of the DC to be able to activate either CD4 or CD1 T cells, which will then drive a cytotoxic T response against tumors with the hope of actually eradicating it. So although the idea is really innovative, The only problem is this vaccine did not really allow us to reach the hopeful clinical outcome, in the sense that we were able to achieve between 4 and 6 months of improved survival. So you don't want to spend, $100,000 for a vaccine and then survive about 4 to 6 months. You're looking for 2 to 5 years of improvement in survival to be able to say that you have a solid and strong vaccine. So, although we've worked in the past on DC vaccines and we have means to actually enhance the efficacy and therapeutic potency of these DC vaccines, we decided to actually take a more innovative approach and switch from dendritic cells to another type of cell, which we call the mesenchymal stromal cells. So why are we really interested to mesenchymal stromal cells? First of all, these are cells that are easily harvested. So from a single bone marrow aspirate, you can actually expand those cells in vitro and achieve billions and billions of these cells. So they generate high yield, and their culturing system is extremely simple. You don't require any kind of complicated or expensive growth factor. Is just regular media with supplements, standard supplements, and the cells will grow. The variety of, more than hundreds of clinical trials have been conducted with MSCs in various indications, and no one was ever able to report any toxic effect of these cells. They have a great plasticity in the sense that they can actually differentiate into all kinds of, cells, adipocytes, chondrocytes. Some people are able to achieve neurons with them, cardiomyocytes. So this is why we say that they can adapt it to your problem, because you can use them the way you want, depending on the conditions you're providing them with the cells. So these cells have been mainly used in regenerative medicine because they can repair ischemic tissues, for example, in the heart, or skin that has been burned. You could use them as an immune suppressant to be able to actually shut down an unwanted inflammation. Two great examples I can give you are they're used in the clinic for graft versus host disease, so after bone marrow transplant, or lately, MSCs have been used to shut down this unspecific inflammation ongoing into patients in ICU that were actually suffering from COVID inflammation. So they've got a great range of application, but the way I looked at them is mostly to convert them into antigen-presenting cells, into a pro-inflammatory cell. And in the past, we've reached that. I mean, my lab is a pioneer in that development, in that project or subject, in the sense that we have demonstrated that we can engineer those MSCs to overexpress, for example, different parts of proteasomal complexes, and these cells will now behave as antigen-presenting cells. Or you could actually treat them pharmacologically and convert them into APCs, like they have a function like dendritic cells, for example. So it's feasible to actually work with these cells and push them to go down a specific road depending on the product you have. So the way we wanted then to pursue that project, we said to ourselves, okay, so we have this accum technology. Our CTSO can generate hundreds and hundreds of molecules of accum variants. So we've got libraries of these molecules. So why not conduct a small high-throughput screening assay where you would take, for example, a mesenchymal stromal cell, and then you would pulse it with two different approaches. The first approach, I mean, we've used ovalbumin as an antigen because we have a T cell that responds to the syncytial peptide, which is derived from ova. So the first approach we did is we would use, for example, ova-linked accum variants versus ova admixed with accum variants. So it's either chemical linking the accum molecules onto the ovalbumin protein, or mixing those chemical variants, these small molecule variants, with the ovalbumin protein. And the difference between these two approaches is that one is much simpler because it really simplifies manufacturing. So the idea would be to pulse these MSCs with the different libraries, and then if the protein is processed, you would have the syncytial peptide on the cell surface. The B3Z T cells will be then added, and then through the quantification of the beta-galactosidase enzymatic reaction, you can then screen and identify the group that was able to activate those B3Z. So this is a very simple assay, cheap, and can give you data within 48 hours. So when we screened using our system, the ova-linked accum variants, we were not able to find any molecule that was able to convert MSCs into antigen-presenting cells. So this is the negative control, so the MSCs alone versus the syncytial pulsed MSCs. You see, this is the kind of signal you'd expect, but really nothing stood out from the screen. However, when we treated the cells with ova that was admixed with the different accum variants, even though we have some molecules that started showing some trends, one of them actually stood out, the A1 molecule. So this molecule here was very interesting because it actually led to a signal that is somehow comparable to the positive control. This is one of the strongest signals you can get from the screen. So taking that molecule now, we then asked ourselves, I mean, very simple questions. Does it enhance antigen uptake and or antigen processing? So usually when you have an antigen presenting cell, the cell will capture the antigen and then it will break it down into peptides to be able to present it on the cell surface. So to answer the question of uptake, we took those ARM cells, so A1 reprogrammed MSCs. We treated, as a negative control. We took regular MSCs. So if you treat them, if you take the ARM cells, if you treat them with the ova that is fluorescent, you have a signal compared to the negative control. If you treat them with A1, you don't get that much of a signal. But if you mix ova AF467, which is the fluorescent ova, with A1, then you can appreciate the fact that the signal is actually much higher than the positive control. So there is an increase in the uptake process, and that also translates in an increase in the processing. So breaking down of the protein, because again, you see here that the signal we're getting with the ova DQ, which fluoresces once it's actually cut, is much higher than just pulsing the cells with ova DQ. So this was actually one of the first impressive data we were able to get. Then we asked this, another question. We know that accum usually can break down the endosome if it's actually linked to a protein. If it's delivered on its own, usually doesn't do much. So the next thing we asked is, does the A1, even though it's mixed with ova or with an antigen, can break down the endosome? So we answered that question using a simple assay. So we took our cells and we added to these recombinant cytochrome C. So cytochrome C is one of the initiators of apoptosis or cell death, because once the protein reaches the cytoplasm, it cuts caspases that will then turn on cell death. So if you don't have any A1 molecule, the cytochrome C will end up being entrapped in the endosome. Eventually, it's going to be degraded with the acidification and the activation of proteases, and the cells will remain alive. Now, if you add in the A1 to that mix, what you expect then to have is to initiate endosomal escape. So the membrane of the endosome is broken down, cytochrome C is released in the cytoplasm, and therefore you end up killing the cell. And that's exactly what we got. So what's interesting here is that if you basically add accum, which is the initial molecule, with cytochrome C, you don't really see a shift compared to the negative control. So there's not much happening. But if you actually mix A1 with recombinant cytochrome C, then you can see that the majority of the population was actually becoming positive for an XN5, which is a signal that apoptosis has already taken place. So this is a nice differentiation experiment, because it shows you that although A1 is a derivative of accum, it does something that accum cannot do when it's actually mixed with an antigen. So now we know that we can enhance uptake, processing, and we can induce endosomal escape. So the next thing we asked is, can this molecule actually trigger reactive oxygen species? So why is this very important? Because reactive oxygen species have been reported in the past to be really important in degrading the endosomal membrane through a process called lipid peroxidation. So it's kind of a mechanistic type of approach to be able to see whether A1 triggers ROS, and that could be responsible for the enhanced antigen cross-presentation. So what we did in this case is that we treated, for example, A1, and we saw here an increase in ROS detection in the cytoplasm compared to, which is comparable to DP44MT, which is a positive control. It's a molecule that does cause ROS. And if you mix it with ova again, you can see that you can also trigger ROS. So that was an interesting observation. The next question we asked is, where is this ROS source? So what we did in order to answer this question is we treated the cells with 3 different antioxidants. So alpha tocopherol is a vitamin E derivative that will block lipid peroxidation, so will block this process of endosomal release. You have mitotempo, which is an inhibitor of ROS production from the mitochondria. And we have NAC, which is a generic donor of cysteine, which is involved in generating glutathione, and that as itself on itself could act as an inhibitor of ROS. And of course, we used an antigen cross-presentation assay to be able to demonstrate whether our molecules can actually block the endosomal escape and have an impact on T cell activation. What's really fascinating here is that if you add alpha tocopherol, which again blocks lipid peroxidation and endosomal release, you actually have a significant decrease in antigen cross-presentation. On the contrary, if you add mitotempo, which acts mainly on the mitochondria, you don't really see that decrease. So that suggests that mitotempo has no, or the mitochondria has no role in producing ROS. And the reason why this is very important is because reactive oxygen species in a cell really comes from two different sources. The NADPH oxidase, which are usually on the cell surface and within the endosome, or the mitochondria, which, as you know, is involved in respiration of the cells and generation of ATPs, for example. This is the electron transport chain. So the next thing we did after that is we said, okay, if we expect the ROS to be derived from the NOx, so what about inhibiting those NOx activities? So to do that, we use two different inhibitors, DPI, which is a generic NOx inhibitor, and ML171, which is an inhibitor of NOx1 specifically. And as you can see here, it's really when you add the DPI that you're able to really decrease the ability of the cell to cross-present. Similarly, you can have a specific, or it's not that powerful, but you still have an inhibition going on, meaning that there might be multiple NOxes that are actually responding to A1. So this actually brings us another level of mechanistic understanding, because it shows that now this molecule can enhance uptake, processing, and it triggers endosomal release by activating the NOx, which triggers eventually or produces reactive oxygen species. Not only that, but we've conducted a more wider screen of the potential effect of the A1 on these cells by conducting what we call transcriptomic analysis. So we looked at all the genes that are expressed, whether it's upregulated or downregulated. And you can say with confidence right now that we have more than 1,500 genes that are modulated in these ARM cells in response to A1. And when we started really looking a little bit more in detail on the different signatures, molecular signatures that could be turned on, we found a variety of different processes, but what really attracted our attention is a process called unfolded protein response. So the unfolded protein response is quite impressive in this context because it's a mechanism of defense of the cell that the cell actually triggers when you have, for example, protein aggregating together, serving as a toxic component to the cell. So the cell produces a variety of different proteins in order to be able to get rid of those aggregates and destroy them. And usually it does that by targeting the complex to the proteasome. So with all these in vitro and mechanistic investigations, we then asked the simple question. I mean, this is where all of our work will converge. Does this ARM vaccine, can this ARM vaccine treat tumors that are pre-established in animals? So what we did to answer that question is we took mice, we transplanted them with EG7 tumors. So these are T cell lymphomas that express the OVA antigen. And then we used the Syngeneic vaccine. Syngeneic is the same thing in clinic as autologous. So it's coming from the patient to the same patient. So this is kind of a personalized approach. So we took that vaccine. We first transplanted the tumor. We give the vaccine after palpable tumors were actually detected, alone or in combination with the immune checkpoint anti-PD-1. So this is the immune checkpoint that is commonly used in the clinic for a variety of indications, including melanoma. So what was really interesting is that if you combine the ARM vaccine with anti-PD-1 in a syngeneic setting, you could actually control very well tumor growth. So, and that leads eventually to 80% survival over 40 days. So this is encouraging because it shows that the vaccine not only works, but it synergizes with the PD-1, because when you deliver it alone, as you can see here with the black dotted line, well, it does delay tumor growth, but it wasn't that effective. Having said that, we asked ourselves, because our primary objective is not to develop a vaccine for every patient, which is in a personalized manner. The idea is to come up with what we call a universal vaccine. So one vaccine to treat everyone. So this is where we started thinking about allogeneic vaccination. So allogeneic vaccination means you take a healthy donor, you generate the MSCs, you convert them into arms, and then you can use that to immunize any non-related patient. So the idea here is that this allogeneic product is going to activate the immune system of the endogenous patient, the endogenous immune system of the patient, and that is going to create a non-specific pro-inflammatory response, which will help during the generation of the immune response against a specific antigen. So it's something that's becoming very common in the clinic. And of course, we used exactly the same setting. And here, what was really impressive was the fact that if you deliver the arm vaccine alone without the immune checkpoint blockers, you can see here through the purple line that we controlled very well tumor growth. In fact, we ended up reaching 60% survival up to 40 days. Now, having said that, if you add in the PD-1 antibody on top of the vaccine, well, there you actually cure these animals. So in fact, eight or nine mice out of 10 in this group actually had their tumors completely gone. And they were 100%, and we achieved 100% survival over 40 days. In fact, this experiment was continued over 3 months. So we went up to more than 90 days, and we couldn't actually see any tumor regrowth. So that was really impressive, because now we have an evidence that the arm vaccine could be used allogeneically to cure solid tumors. And additional investigations are actually undergone right now to be able to use, for example, B16 lysate, which is a melanoma model, to demonstrate that we can use lysate to cure, a specific tumor, a specific melanoma tumor. And the idea here is that if you want to treat a variety of different patients, you have to use their own tumor cells, their own lysate. So this is why we're going to call it a universal semi-personalized vaccine, where the cell is universal to everybody, but the tumor lysate has to be coming from the patient himself to be able to activate the immune system against their own neoantigens. So overall, if I may just summarize what we were able to show today, by mixing an antigen with A1, we're able to actually induce or activate the NOx, which will generate reactive oxygen species. These ROS will destroy the endosomal membrane, allowing the product or the antigen to be released, which will then be targeted to the proteasomal complex for degradation. The peptides will be loaded on MHC class 1 and be presented on the cell surface. Now, what's really interesting, I told you about the unfolded protein response. So we think, and that's a work in progress, that mixing an antigen with A1 will cause protein aggregates, so a lot of large complexes, which will then, when they are released from the endosome, will serve as a signal, as a danger signal to the cell to target that complex to the proteasome for degradation. So this is something we're trying to work on right now to demonstrate through turbidity assays that these aggregates are there, and that's what really pushes the complex to move on to the proteasome. So as I mentioned earlier, there is an ARM phase 1 being generated right now, prepared. So it's a phase 1/2A trial, 3+3 design. The idea is to find the most effective dose and then test that dose with an immune checkpoint blocker so we can basically look both at safety and at the same time eventually demonstrate some sort of efficacy to move on later on to a phase 2 trial. So I hope I was able to actually convince you that the accum technology could be used heavily to not only generate, protein-based vaccines or enhanced ADCs, but also in the generation of a cellular vaccine, which, by the way, could be applied to any kind of tumor, given the fact that you have access either to a tumor-specific antigen or tumor lysate. So if you have further questions in the future, please feel free to communicate with us or communicate with the CEO at the following email address, and we'll be more than happy to answer your questions. Thank you.
Very interesting presentation, Dr. Rafi, as usual. Julian DeRosie here, taking over moderation from Matthias. Thank you, Matthias, for your work. Maybe, Doctor, first question, the Biden Moonshot Cancer Initiative, how can that impact your business going forward?
Well, the Biden Moonshot Initiative is actually to promote, treatments for cancer. So the idea here, the timing is perfect because that's what we're trying to do. We want to actually reach for investments. We want to try to push our vaccine to be able to treat not only melanoma, but a variety of different indications. And I think, with the awareness that is being provided right now to different people, to the world, I think that could be actually a good promoter of our technologies and our advancements in the field.
Absolutely. And Jeff is asking, my understanding is that A1 is a derivative of accum. If so, what is the derivation?
Well, as I mentioned earlier, the accum is basically composed of the bile acid and nuclear localization signal. So what we've been doing is we've been changing these components. So there are a variety of different bile acids, there are a variety of different peptides we can use. And it's with these combinations that we're able to actually generate a variety of molecules. And when we screen them, that's the one of these molecules actually was able to turn on the antigen cross presentation. Now, for confidentiality purposes, I cannot really say exactly what these sequences are, but you know, through CDA or NDA signatures, we can then move on and disclose this information later on.
Perfect. Well, time is up, Dr. Assay. Thank you very much again for your presentation. Very interesting. We obviously encourage you to follow Defense Therapeutic Stories. Both get in touch with the CEO or Dr. Assay. And now it will be time to move on to Hanova Reinsurance. So thank you, Doctor.
Thank you. Have a great day. Bye-bye.